I am postdoctoral training in bioinformatics. Her main research activities focus on the gut microbiota in various diseases, with emphasis on dietary interventions and the microbiota gut-brain axis. Her current projects combine metagenomics with other omics data, such as metabolomics and proteomics. She uses machine learning algorithms to detect health or disease-associated signals in big data. And her main focus is a cohort of children with severe drug-resistant epilepsy that are treated with the ketogenic diet. And that is going to be the focus of her talk today. Thank you again, Dr. Prast-Nielsen, for spending time to share with us your expertise. If you would like to share your screen.
So you see the main slide, right? Okay. So I'd like to thank you for inviting me and giving me the chance to tell you about gut microbes, because that's probably not one of your most common topics in this teaching network, but I do think it's very important that neurologists are aware of the microbiota. So I will talk about epilepsy, the ketogenic diet, and the role of the gut microbiota. My disclosures, I have several funding agencies, but otherwise I have nothing to disclose. The learning objectives for today are that after this lecture, I hope you understand the role of the gut microbiota for human health, and you're able to reflect upon the gut microbiota's role in neuronal activity and epilepsy, and are able to discuss the modes of interaction of the gut microbiota with medication, and summarize the current knowledge about anti-seizure medications and the gut microbiota. And I can give you a spoiler here, there is not much knowledge, so I think you'll remember what is known about those. You should also be able to describe the role of the gut microbiota during ketogenic diet treatment and identify important current knowledge gaps to be addressed in future studies. So I would like to start with introducing the microbiota to you, as this is not the field of your research. We talk about the microbiota when we talk about the microbial community that is living inside the human body or on its surfaces, and contains bacteria, virus, fungi, all commensals, the good bacteria and pathogens. And when we talk about the microbiome, we usually refer to the collective genomes of the microbiota, but the microbiome can also be the environment the microbiota resides in. What is really
Dr. Nielsen, sorry to interrupt you, but we are seeing your notes view of presentation.
Is this better?
Yeah, we still see your upcoming slides on the bottom. So
wait, then we tried this before. Oh, here, let's see.
Yes, I think
this
is
okay?
the full screen mode. That's
yes.
Sorry for the interruption.
Yeah, no problem. Thanks for letting me know. Yeah, so we really need to remember that humans are basically holobionts. We are one microbe that interacts with millions of microbes. And the composition of our microbiota is influenced by birth mode, diet, and medication. The main functions of our microbiota are to defend us against harmful microorganisms, teach the immune system and train the immune system, and the microbiota produces important metabolites that also, when they're produced in the gut by the gut microbiota, they still can influence the periphery by traveling through the circulatory system to different organs. And to give you an idea about the size of the microbiota, we know that there are as many bacterial cells present in our microbiota than there are human cells in our body. So on a cellular level, we're about 50% human. However, since the microbiota can contain up to 1,000 species or more, and they have different genomes containing different genes, it has been estimated that our microbiota encodes for around 3 million genes, which is much more than the number of genes in the human genome, that is around 23,000 genes. So on a gene level, the microbes really outnumber the human genes. And 90% of our bacteria are located in the large intestine, which is one of the most densely populated microbial ecosystems on Earth. And since the composition of the microbiota is heavily influenced by different environmental factors, every individual has their own microbiota. So there is not two persons on the planet that have exactly the same composition. And let me see. Yes, we talk about the dysbiosis when we see a disruption of the microbial homeostasis, where beneficial microbes are diminished and potential harmful ones may outgrow. And this has been shown to contribute to a range of different diseases, not only in the gut, where we have inflammatory bowel disease or colorectal cancer as examples, but a dysbiosis in the gut can also be associated with other diseases, including neurological disorders like Alzheimer or Parkinson's. And how can the microbes in the gut influence our brain, you may ask? So there are different routes of communication. They're usually summarized as the microbiota gut-brain axis, and this includes different mechanisms. There is the metabolic route of communication that includes neuroactive bacterial metabolites that can be taken up by the circulatory system and reach the brain. This also includes stimulation of the vagus nerve. For example, lactobacilli are known to stimulate the vagus nerve. The microbiota, as I mentioned before, stimulates the immune system. Depending on the species, they can induce pro or anti-inflammatory cytokines, and the endocrine system can influence the composition of the gut microbiota. So different ways of communication between the gut and the brain, and the dysbiosis in the gut that I mentioned before, when there are potential harmful bacteria that can outgrow, this can lead to increased oxidative stress and the damage to the gut barrier. And this can also cause an inappropriate immune response and the damage to the blood-brain barrier. And in worst case scenario, this can cause neuroinflammation, and this may impact seizure thresholds. The gut microbiota is not only important for adult brain function, it's actually also really important for brain development. And here studies of germ-free mice have been really vital to learn about this. Germ-free mice are mice that are born and raised under strict sterile conditions. They never have a microbiota. And comparing those to normally colonized mice, it was shown that brains fail to develop normally. Some examples are that there is some increases in hippocampal and amygdala volume, morphological changes in different parts of the brain, increased adult neurogenesis and hypermyelination have been recorded as well as differential expression of several genes that are involved in fundamental brain processes. And these are ranging from neuronal plasticity to metabolism, neurotransmission, and morphology. And I list a few key papers here if you want to dig deeper into this subject. I also would like to really recommend a review article by Dodge and McCafferty that was published last year with the title Gut Microbiome Effects on Neuronal Excitability and Activity, Implications for Epilepsy. And they really did a nice job summarizing all the current papers that have been published. And here I'm showing just the two first tables where the studies are summarized for how the microbiota impacts neuronal excitability and neuronal activity. And as you can see, most of the studies were done in mice, and these down here are just some human studies. But most importantly for this talk, I think, is the summary in this table, the microbiome effects on epileptic neuronal activity. As you can see, all these studies are published in 2018 or later. So this is a really new field, and there's really still much to explore. And I would like to highlight those two oldest papers here. Olson et al. I will talk about when I talk about the ketogenic diet. And Middle-Martis et al. I would like to highlight here in the figure to the right. The experiment they performed was that they used two groups of rats, and one group was chronically stressed 2 hours per day for 2 weeks, and the other group was not stressed. And then they recorded seizure propensity upon kindling, and the chronically stressed rats, they showed accelerated progression and increased seizure duration compared to the sham stressed rats. But then they collected the fecal samples from those rats, and those contain the gut microbiota, and they donated them to the other rats. So sham-stressed or non-stressed rats that received a fecal microbial transplant from the stressed rats, they also showed accelerated progression and increased seizure duration. And the stressed rats that received the fecal transplant from the non-stressed rats, they became less seizure prone. So here they could actually show that the seizure propensity was transferred through the gut microbiota. A very important finding. The same group also published another article just last year with the title "Susceptibility to Epilepsy After Traumatic Brain Injury is Associated with Pre-Existent Gut Microbiome Profile." That is also a very interesting topic. As I said in one of the first slides, the composition of the gut microbiota can be modulated by medication and diet. I'll talk about medication now and diet after that. But also the microbiota can actually impact drug response. This can happen by different ways. So if you remember, I said we have about 3 million microbial genes that potentially interact with us. And it is thus not unlikely that a drug, especially orally taken drugs, that a drug can be metabolized by any of those microbes in the gut. And this can either lead to an activation of a prodrug, an inactivation of an active drug, or that some microbes can actually metabolize a drug into a toxic metabolite. On the other hand, the drug or metabolite can have antimicrobial effects on the gut community, or it can induce selective bacterial growth. And this antimicrobial activity, now we're talking about non-antibiotic drugs. We're talking about drugs that have human targets. But this antimicrobial activity among these drugs is actually not uncommon. There was a paper published in Nature a few years ago where they could show that 24% of all drugs across all classes with human targets inhibited the growth of at least one of 40 representative gut bacteria strains. This was a study that was performed in vitro when they screened over 1,000 different drugs. And again, 1 in 4 of those drugs impacted the microbial growth. And as I said before, the composition of the gut microbiota is important. So this can actually impact health and disease. In addition, in this study, unfortunately, they did not look at anti-seizure medication. In addition, gut microbes can also influence drug transport, and this was in connection to epilepsy, recently shown in a paper published in 2022, where they could show that Bacillus subtilis, a common gut microbe, could actually inhibit the ABCB1 transporter that can transport anti-seizure medication across the intestinal epithelium. So what do we know about gut microbial interaction with anti-seizure medication? One study was published last year where they did a similar screen as it was done in the Nature paper. And so it was in vitro using 32 representative gut microbes. And they tested 9 anti-seizure medication active ingredients, 7 common excipients, and 4 syrup formulations. And the main findings were that carbamazepine, lamotrigine, and topiramate, as well as propylparaben, which is a syrup, reduced the growth rate of more than 10 bacteria strains. And in this figure up here, the bacterial strains, they are all summarized at higher taxonomic level. But what this figure is demonstrating is that carbamazepine has a different spectrum or antimicrobial spectrum than lamotrigine. For carbamazepine, bacteroidetes and verrugomicrobia were most sensitive, whereas lamotrigine had its most inhibitory effect on actinobacteria. And to the group of actinobacteria, for example, belong the bifidobacteria that I'm sure many of you have heard of before. They could also show in this study that artificial sweeteners present in anti-seizure medication formulations could stimulate the growth of certain gut microbes. They can basically use this as an energy source. Then there was one more paper on the subject published in 1979, where they could show that zonisamide was actually metabolized by typical intestinal microbes, including Bifidobacterium bifitum. Other than that, we don't really know anything about how anti-seizure medication and the gut microbiota interacts. We don't know anything about this in patients, actually. So we really need large patient cohorts and well-designed studies to study this. And the composition of the gut microbiota may be important for the therapy response. I mentioned that diet also shapes the gut microbiota. I will give a short introduction on diet in general before I come to the ketogenic diet. So the normal diet usually contains mostly carbohydrates, and then we have fats and proteins. And complex carbohydrates are the most important energy source for the gut microbiota. This in the figure to the right is represented by our little friend, the broccoli. We don't have any own enzymes or transporters that can actually metabolize and take up those complex fibers in our small intestine. So when we eat those, they will travel all the way to the colon, where most of the gut microbiota resides, and they can metabolize those fibers, and they can produce, for example, short-chain fatty acids. And these have anti-inflammatory and anti-carcinogenic properties. They can stimulate the immune response, and they act as epigenetic modulators as HDAC inhibitors, even in the brain. They can reach the brain, they're quite small. However, a typical Western diet these days does often not have that much complex fibers. Much of the carbohydrates consist of more simple sugars that are represented by the little piece of cake here. And those can be metabolized and taken up into circulation by our own genes in the small intestine. If most of our carbs are those simple sugars, there is not much carbon energy sources reaching the colon, and this will starve our microbiota. And this can lead to the outgrowth of other microbes that are not highly dependent on those complex dietary fibers. These are microbes that can either digest dietary proteins or endogenous energy sources, which includes dead cells in the gut, and importantly, the mucus, which is a protective layer between gut lumen and the gut epithelium to protect us from pathogens. So those microbes can digest this mucin and diminish this barrier, and this will make it more easy for pathogens or other harmful bacteria to get into direct contact with the epithelium, and this can cause inflammation and the so-called leaky gut. In addition, the Western diet often contains quite a lot of protein. This can be fermented into amino acids by the gut microbiota, and this leads to the production of pro-inflammatory and cytotoxic metabolites. And it has been shown that these can contribute to chronic diseases like colorectal cancer. So we know most about the carbohydrates, how they actually interact and how important they are for the gut microbiota. We don't know that much about the fats, and we know a little bit about the proteins, but we know that those fibers are, they are important for gut health. So then I'm sure you're all quite familiar with the ketogenic diet. I just quickly recap, it's a high fat, sufficient protein, very low carbohydrate diet. And if you don't actively supplement with any fibers, this is also a low fiber diet, because the fibers are usually in natural foods. They're usually mostly found in grains and fruits and vegetables that also contain other carbohydrates that you're trying to exclude in this diet. And when you're on the ketogenic diet, you experience a metabolic shift in the brain from glycolysis to metabolism of ketone bodies. And this can reduce seizures in about 50% of drug-resistant patients, but it's currently not really known why some patients respond to the diet and why others don't. And this can currently also not be predicted before a patient is treated. And there are many hypotheses on how the ketogenic may decrease seizures, which I've listed here, which you probably know most of them. But I do want to add and emphasize the change in the gut microbiota composition here. And this can also be part of the reason of the anti-inflammatory actions or the epigenetic changes, for example, that can lead to this, to the positive therapy response in the ketogenic diet. Before talking about our patients in our own study, I really want to highlight the paper by Olson et al. That was published a few years ago about the ketogenic diet study in mice. They used 2 different epilepsy models here, and they combined metagenomics with metabolomics in the blood and in the brain, and they could disentangle mechanism how the ketogenic diet actually can lead to less seizures, where they showed an increase of specific microbes in the gut, which was associated with a decrease in gamma-glutamylated ketogenic amino acids in the blood and an increased GABA and glutamate ratio in the brain, leading to less seizures. This mechanism, we don't know yet today whether this is actually happening in patients as well. In our study, we have a metabolomic data set that we are currently analyzing, and also we will be specifically looking at those ketogenic amino acids. However, what's really important in this paper as well is that the mice, when they were on a ketogenic diet, they had less seizures, but mice that were germ-free, as I said before, they were sterile, they did not have a microbiota, then their seizure threshold was not changed by the ketogenic diet at all. But when those mice then were introduced a microbiota, and then the ketogenic diet had an anti-seizure effect. So their conclusion was that the gut microbiota mediates the anti-seizure effects of the ketogenic diet, and at least in the mice. We still don't know whether this is true in patients. So there are several studies out there that have looked at the gut microbiota during ketogenic diet in patients with different tools. And unfortunately, all of those studies have very small heterogeneous cohorts, and our study is quite small as well. But it's difficult to get like large numbers of patients for this. But what makes our study a bit different from others is that we used whole metagenomic sequencing of the fecal samples, which allows us not only species resolution of the microbes. The other commonly used tool is 16S analysis, where you don't get this species resolution. And in addition to the species resolution, we also can analyze functional profiles of the gut microbiota. And this study was published in 2019. We had a small cohort of 12 children with drug-resistant epilepsy. This was a collaboration with Dr. Maria Dahlin from Astrid Lindgren's Children's Hospital here in Stockholm. And we could see that during the ketogenic diet, mainly bifidobacteria disappeared. And we saw that E. coli increased during this treatment. And by looking at the functional profiles, of the ketogenic diet, of those samples, we could see that carbohydrate metabolism was significantly changed, and this was associated. The genes for the carbohydrate metabolism, they also decreased during the ketogenic diet, and this was associated with the decrease of the bifidobacteria. So these were functions the bifidobacteria would carry out in the gut. We then continued our study with a slightly larger cohort, still not that many, but we're trying. We used the machine learning approach to analyze the composition of the gut microbiota, and we could confirm the major changes that E. coli increased and bifidobacteria decreased. But here we also had serum samples where we did we ran an inflammation profile of 92 different inflammatory peptides. And you are not able to read all those peptides here, but you can look up the publication for that. But we could basically see all those red dots here are inflammation markers that were significantly decreased during the ketogenic diet. 3 of them increased, but the majority decreased. So in general, the ketogenic diet had an anti-inflammatory effect in serum. But then we also wanted to compare the responders to non-responders in this cohort. We had about 50% responders and non-responders. And we could see before starting the ketogenic diet, the responders had a significantly higher inflammation, basically mainly 3 important inflammatory molecules, which were TNF, IL-12B, and MCP4. And during the ketogenic diet, there was no significant difference between those patients. So it seemed like the non-responders, they started on a slightly higher inflammation. And both TNF and IL-12 significantly decreased during the ketogenic diet. For MCP4, we only saw a trend, or well, it was most significant. But we've further looked into this inflammatory profile and the metagenomics profile by integrating these 2 omics data sets. And we could see that both Bifidobacterium breve and longe correlated with serum TNF. And this was only true in responders before the diet. So here we have the TNF, and the purple lines are positive associations with all the different bifidobacteria that you can see here. There are also some other inflammatory peptides that positively correlated with the bifidobacteria, but to a much smaller extent. The most significant, the strongest correlation was only with the TNF. And we saw that the correlation was only with some bifidobacteria, not with all bifidobacteria in the data set. So we performed some co-occurrence clustering of all our bifidobacteria, as a matter of fact, of all of our bacteria in the data set. And we could see a clear clustering of the bifidobacteria into 2 different clusters, one in light blue here and one in white. And the one in light blue is infant-type bifidobacteria, and those are all the bifidobacteria that actually correlated with TNF. And the other bifidobacteria that did not correlate with TNF, they were in a separate cluster, and they are more adult-like bifidobacteria. So when a child is born, one of the first colonizers of the gut are bifidobacteria, and they reach a peak within the first few years, and then they decrease. So there's a negative age correlation. Our patients were at least 2 years old, and from that point you expect a negative age correlation in a healthy child. So we looked at age correlation in those infant-type bifidobacteria, and we could see this negative correlation before the diet in the patients that were non-responders to the diet. However, this negative age correlation was not existent in the responders. So even as they got older, they still had quite a lot of those infant-type bifidobacteria. But when starting the ketogenic diet, those bifidobacteria decreased. And 3 months after the ketogenic diet, when we collected samples again, there was a negative correlation in both responders and non-responders. And we put our findings into context. We could find that TNF was highly dependent on intestinal B. longum, which is an infant-type bifidobacteria, but not B. adolescentes, which is an adult type bifidobacteria, in a large Tanzanian and Dutch cohort. This was recently published. And what we think is that for some reason, those patients that respond to the diet, they have a more immature gut at older age. They still have more infant type bifidobacteria as they should have. And a similar finding was published by Wan et al., where they looked at children with an autism spectrum disorder diagnosis, where they could show that they had persistent underdevelopment of the gut microbiota compared to neurotypical age-matched peers. And ASD and epilepsy are common comorbidities, so there may be a role for the bifidobacteria here. So to summarize our findings here, we see an increased levels of infant-like bifidobacteria and tumor necrosis factor in children with drug-resistant epilepsy, and they are associated with anti-seizure response to the ketogenic diet. But this study needs further validation in larger cohorts, including adult patients. And either TNF or bifidobacteria could be used as biomarkers to predict anti-seizure response prior to ketogenic diet treatment, and targeting these bifidobacteria then might become a novel anti-seizure therapy. To further support our ideas about the bifidobacteria, there was another paper published by Lack et al. In 2020, where they studied mice that were neonatal mice that were colonized with 4 infant-type bifidobacteria species, and they were compared to germ-free sterile mice. And they could show that those with the infant-type bifidobacteria showed increased TNF expression. So this supports our finding. They could also show decreased expression of synapse-promoting genes and increased microglia activity, increased Purkinje cell firing rate. However, they did not really study the communication of the gut-brain axis in this study. Ando et al. Also showed a few years ago or proposed that synaptic pruning by microglia is enhanced in the epileptic brain, and Cross et al. Demonstrated seizure-related changes in Purkinje firing frequency. So we think there may be a role potentially of infant-type bifidobacteria in seizures. And decreasing those through the ketogenic diet can have a positive effect on seizure thresholds. So this was our study on the ketogenic diet. We're still continuing. As I said, we have a metabolomics data set that we're currently analyzing, but we're also currently planning on a much larger study, because as I said, most of the studies that have been published on the gut microbiota and epilepsy and the ketogenic diet, they're really small. And we need really large patient cohorts to actually make really any good conclusions. And we think that the gut microbiota can have an impact on seizure thresholds, and it may be important for some epilepsy syndromes, but maybe not for all of them, maybe mainly for those that are associated with an inflammation that can be potentially caused or exacerbated by the gut microbiota. So we're currently planning a large study where we recruit treatment-naive patients, all different kinds of epilepsy diagnosis, in order to study which types of epilepsies are actually associated with changes in the gut microbiota. This will be fundamental for more focused studies in the future. But we also want to follow up those patients during their first anti-seizure medication treatment to study which anti-seizure medication microbiota interactions may actually exist in vivo. And we are currently focusing on major hospitals in Sweden to recruit patients. We've also started making contacts to other Nordic countries in Europe. But I think this is a study that can be potentially be global. So if anyone listening is interested in participating or wants to have some advice or support for setting up such a cohort in their respective country, you're all welcome to contact me, and we can see what we can do. So to finish up this talk, my take-home message for today are that the gut microbiota has a significant impact on health and disease, including neurological disorders. The gut microbiota may impact anti-seizure medication and ketogenic diet treatment outcome. Further studies are needed to identify specific microbes and their mechanisms, and modulating the gut microbiota might become a novel anti-seizure treatment alternative. In the end, I want to acknowledge my collaborators, both here in Stockholm, but also at George Washington University in the US, and VMI, and my collaborators there. And I want to finish with a picture of Stockholm and as what it usually looks like in February, March, this time of year. And I want to thank you for your attention. And
thank you very much.
Yeah, I'm looking forward to some questions or discussions.
Yeah, there are already some questions in the chat, so I'll read them for you. First one is ketogenic diet has low fiber content, hence it should theoretically increase inflammatory mediators, but studies show that the ketogenic diet could reduce inflammatory markers. Why is that?
That is a very good question, and we don't really know the answer yet. I think it's maybe due to that the patients that start the ketogenic diet are not healthy. They may have a high inflammation, and in a healthy person, the ketogenic diet may have a different effect than in a patient. We have not included, in our study, we have not included any healthy individuals undertaking the ketogenic diet treatment. That is, of course, a weakness in the study, but I would not recommend this diet to anyone healthy, to be honest. So
the next question is, sometimes we see change in seizure frequency associated with short-term antibiotic use. Could it be due to change in the gut microbiota?
It could, there are different, there is one small paper, I think with 6 patients, where they look at that, and their conclusion is that it could be due to the change of the gut microbiota, but it's difficult. Those patients, they also have an infection. So you don't really know what is changing what in this perspective, but antibiotics have a big impact on the gut microbiota, and this could potentially have an effect on seizures, yes.
Does the vagus nerve stimulator have any effect on the gut microbiota, which in turn may affect seizures?
This is something I'd love to study. Here at our hospital, we have too few patients that actually start the VNS, start on the VNS, because you need to have a sample before the stimulator is used and then during this, the treatment. But if there is anyone out there that sees lots of patients that is interested in setting up a study, I'd love to be part of that, because I think there is something interesting to find there.
Okay. There's a request if you could put up again your slide with your email contact. I think there are people interested in making sure they annotate that. Or
it's also sure, I'll share it again, but it's also it's recorded, right? Yeah, you can find it. Oh, sorry.